Start recording. All right. Okay, friends. Oh, yeah. 121 to 124. 121 to 124.
Oh, shoot.
Oh, shoot. Yeah. Okay. Okay. All right. I thought today we might actually go back in time, and I just have to find where it is. I think it's this one. Yes. So let us go back to 5.2c, page 34, because there's a few things in there that I just want to make sure that we have covered, and we just kind of skipped over it. I let you read it on your own, but I want to make sure that you know all about it. So this is page 34. So this is all about different types of chromatin and chromosomes. So DNA. DNA is that double helix shape. And then what does it do? It coils around, what do we call those little things? The histones. You can either call them histone proteins or you can call them histones. And once it has coiled around those histone proteins, it actually sort of spins itself kind of like that, and it becomes chromatin. And this is the form that I like to call the long, thin, and my favorite word, noodly. Why is it noodly? Because it is like, page 34, 34. Why do I call it noodly? Because it is like noodles. Okay, it is long and thin and noodly form of DNA. And that is our chromatin. And in the diagram, you can see the chromatin. Lucas? You can see the chromatin, but in reality, can you see the chromatin in the real picture? Anybody see anything that looks like noodles over here? Do you see noodles or no noodles? No noodles. And that's because DNA in reality is so thin you cannot see it. So even though in pictures we will draw it where it looks like you can see the noodles, you cannot actually see it. Not unless you get in there with like an electron microscope. Okay. So there are two ways to go about this. We can either start from the big structure. This is the coiled up replicated DNA. What do we call that? The coiled up replicated DNA is called a chromosome. What do we call the two different strands, the two sides of the replicated chromosome? The two sides. This is one we just learned yesterday. Anyone know it? They are chromatids. They are called sister chromatids. Chromatids. Are those sister chromatids similar to each other, different from each other, or exactly the same as each other? They are exactly the same as each other. So if you take that those sister chromatids, those replicated chromosomes, that coiled up chromosome, and you unravel it, by the way, what is the word meaning coiled up, condensed? The unraveled form, what do we call that? That is chromatin. And what are those things? Those little circly things? Those are histones. And what do we call this kind of structure here? Beads on a string. And then if we take away the histones and we uncoil it, what do we end up with? We end up with our double helix, which is just the DNA. So if you take away the histones, you've just got the DNA. So you can take this diagram and either read it from most coiled up to least coiled up, or you can take it from DNA and say, oh, it starts getting coiled up, and then it coils up more, and then it coils up even more. Okay? Why does DNA condense? It's so it can be more easily organized. Why would we want to organize it? For what purpose? Because you know what? It's really good at doing its job without any organization whatsoever. You can read it, you can make RNA copies, it can make those can go out and make proteins. Why would we care to organize it? What process is going to happen? Cell division. So it can be more easily organized for cell division. Okay. And here are some actual chromosomes under a scanning electron microscope, and they really actually do look like little X's. Okay, that's pretty amazing. But remember, what did it look like before they were all coiled up? Let's go back and look. There's our nucleus. What do you see? Nothing. Just nothing. The dark part is the nucleolus, yes. And what is the purpose of the nucleolus? Remind your partners, what does the nucleolus do? The nucleolus makes something. It makes ribosomes. Okay. All right. Here's DNA during cell division. This whole process here is called if you are interested, I know some of you did this in grade 6 or something like that. I will tell you the different stages of mitosis, but we're not going to get too excited about it. If you're like, I don't want to know the names of it, I just kind of want to know what it looks like, that's fine. So here we have chromatin. You can't even see it. So before a cell divides, the chromatin, actually there's a step in here that's missing. We'll call this 1.5. The DNA makes a copy of itself. Why is it making a copy? You need double the amount. You need one to go to this daughter cell and one to go to that daughter cell. If you don't have that, you're going to end up with cells that just have half the DNA. And then if it happens again, you'll have a quarter of the DNA. And then if it happens again, you'll have an eighth, and then you'll have like nothing left, and that would be sad. Okay, so it has to make a copy of itself, so each daughter cell gets a copy. Whoops, I can spell daughter. Sure I can. I don't know. That's a good question. Daughter cell. Daughter cell gets a copy. Oh, it's so funny. I'm on the timetabling committee, so we like put up the blocks and try and organize them so everybody gets the courses that they want and so on and so forth. And right now we have some jobs where we don't know who the teacher is going to be because, you know, there's either like Mr. Weinberg is retiring, for example. So we have a teacher X, right? We don't know who it is yet. They're going to be hired. But we have little magnets that we put up for them. And I just bring this up because I just made so many mistakes in that sentence. And so when they made like the little sticker to go on there, they wrote, Teacher X. And I was like, we cannot put that up on the board. We are a place of education. We cannot write Teacher X. Anyway, so we made a new magnet. It was fine. Anyway, it was kind of funny. So each daughter cell gets a copy, and I don't know why they're called daughter cells and not children's cells or son cells, but they are called daughter cells. So, can you see them starting to coil up? Do you see how it goes from like, you can't even see it? Whoops. Oy. Do you see how it goes from you can't even see it into being coiled up? Can you see them now? You can see threads, right? That looks really messy. Those are coiled up chromosomes, condensed chromosomes, condensed replicated chromosomes. But you can't really, see individual chromosomes at this point because they're all kind of lying on top of each other. And there's 46 of them, so that's kind of crazy. And now they've finished coiling up. How many sister chromatids each? How many sister chromatids in each chromosome? So, hang on. Chromosome. How many chromosomes? 46 chromosomes. How many sister chromatids in each chromosome? 2. Because here's one chromosome. Yes, and it's made of 2 sister chromatids. So there's 26 of those little ones. There are 96 of those little ones. Huh? Okay, so you got, say this is chromosome number 4, okay? Because each chromosome has a number, okay? There's chromosome number 4 from dad. There is chromosome number 4 from mom. Because they have been replicated, there are 2 sister chromatids per chromosome. Replicated chromosome. So there's one chromosome from dad and a replicated one from mom. Yeah, there's a replicated chromosome from dad and a replicated chromosome from mom. An unreplicated chromosome, we could show it like this. Okay, and we would have unreplicated from mom. Now, to be honest, this is a little bit of a lie because you'll never end up with a condensed, unreplicated chromosome. Replication happens when it is uncondensed because it can't be all coiled up and be making a copy. It's too compact. It needs to be able to open up to make a copy. So the copying, really what's happening is that we have back here, let's just show 2 chromosomes, okay? This is chromosome from dad, okay? From dad. And I can't even put it, well, actually, I could put it in there, I guess. Could I? Let's put it in the nucleus. Okay, that one's from dad. That one is number 4 from mom. And then the DNA makes a copy of itself. So you'll end up with the long, thin, noodly form with making a copy. Okay? And then you have the long, thin, noodly form that makes a copy. But then they coil up into threads you can actually see. Like these would be so thin you can't see. So the copying takes place while it's so thin you can't see it. Then it coils up. Okay, now you have all these replicated chromosomes, 46 of them, each with 2 sister chromatids. And then, step 4, we have to go back up to the top of the page. We have to organize them. And the picture on the left is an actual cell where they are lined up. And you can see actual cells don't look as beautiful as the diagram. Okay, this is an actual cell. That's a diagram. The diagram makes it easier to see, yeah? In this case, how many chromosomes do we have? We've got 4 chromosomes. In this one, 4 replicated chromosomes. Yes. Oh, this is because I cannot draw 46 on a page. We are showing, we're not doing a human cell here. We're doing some animal that only has 4 chromosomes. Okay, super simplified so that we don't have to draw 46. Does that make sense? Okay. These 2 here are the same size. They are the same shape. What do we call these 2 together? What do we call these 2 together? The ones that are the same size, that are the same shape. Those are called what type of chromosomes? These are the sister chromatids. So one came from dad and one came from mom. And the pair that came from mom and dad are called homologous. There's a homologous pair of chromosomes. Do you see how the other 2 are also the same size and the same shape? This is another homologous pair. One is from dad and one is from mom. Yes. Homologous means similar but not identical. Okay? Yes. Yes. Homologous chromosomes are similar but not identical. For those of you who are leaving, no worries. The lesson is being recorded. Do you know where to see the lesson online? I'll show you real quick. If you go to Teams, there's probably some hidden channels. Yours is probably the hidden channels. You'll find something that says lesson videos. Okay, and if you click on lesson videos, you can see that yesterday's video is there, and there was no video on Wednesday, but there's Tuesday's video. So if you're leaving now, have a wonderful time, and I will see you tomorrow. Not tomorrow, because tomorrow's Saturday. I will see you on Monday. Okay? Have fun bowling. We'll give you a minute to sort yourselves out there. Yes? Only 15? To be honest, this time of year stresses me out quite a lot So I'm not disappointed that there's only 15 because I could stand for this energy to like come down a lot. Yeah. I mean, I love teaching. It's an enjoyable thing to do. I like working with you guys and whatever, but like I just need a break. And I feel like you guys are probably in the same boat. Yeah. Yes. No. Chemistry 11. And chemistry 11 enriched. Okay, back here. Let's go. Homologous chromosomes are similar but not identical. So those chromosomes might have the eye color gene, the hitchhiker's thumb gene. Both of them will have those genes. But they don't have to be identical because maybe you got a green-eyed gene from mom and a blue-eyed gene from dad. Yes? Or maybe you got a brown-eyed gene from mom and a brown-eyed gene from dad. Maybe they are the same for some things, but they can be different for other things. Homologous chromosomes have all the same genes. For all the same traits. But they're not identical. Why are they not identical? Yeah, they can have different versions of the genes. Yeah, exactly. The lowercase s and the uppercase s. Lowercase t versus capital T. So maybe you have a hitchhiker's thumb gene, that gives you a capital T. And we don't know whether you have two capital T's, because as long as you have one capital T, your thumb can bend backwards. But if your thumb doesn't bend backwards, that means you got a little t from mom and a little t from dad. Okay, what do we call these different versions of the genes? Those are called alleles. So going back to the cell division process, all of the chromosomes line up across the middle of the cell. What that's going to allow is it's going to allow, you can see in the next step, they start to get pulled apart. There's these little protein fibers that pull them towards the ends of the cell. And the sister chromatids split apart. The identical sister chromatids split apart. Okay, so everybody lines up along the middle. The sister chromatids split apart. at which point they just become a chromosome again. And eventually they all end up at one end or the other end of the cell. Okay, so you can see one set of chromosomes. This is the other identical set of chromosomes. And then the cell pinches in two. And the chromosomes uncoil. And if the chromosomes uncoil, what format is the DNA in again? It's now chromatin, and it's invisible, which is why I did not draw it. Too small to see. Too thin to see. Did that help clarify a few things? Yeah? A couple other things I just wanted to mention while we're here is if we had T, which is hitchhikers, whoops, let's spell that correctly. Hitchhiker's thumb. Straight thumb. If that's what you had, if those were the two alleles you had, we would say that you are what's that word that we say when the two alleles are different? When they are different. What's the prefix that means different? Hetero. They are hetero. And then the last part of that word is zygous. Okay, heterozygous. And what if it was little t, little t? What if we had the same ones? That would be homozygous. All right. And I think at this point, I don't want to do much more because a bunch of people are away, and I think that was probably enough to take in for today. Do you guys want to now add on to your cheat sheets? Does that sound like a plan? We will have a quiz on Tuesday on genes, chromosomes, alleles, a quiz on Tuesday, and then our test will be on Friday. Okay? So quiz Tuesday, test Friday. Yes, cheat sheet for test. Okay? What I would say is, this weekend, if you would like to be an academic student in your grade 10, 11, 12 years, I would study the stuff as though I wasn't going to have a cheat sheet. I would make my cheat sheet, but then I'd be like using it like flashcards and stuff. Okay? Because when you get to grade 11 and 12, you're not going to have cheat sheets. Do you know what I mean? But if you're like, you know what, I am not going to go into biology, there's way too much vocabulary for me, it will drive me crazy, then don't worry about it so much. So it depends on what your goals are, right? Is your goal to go and take those academic courses that require some memorization? Great. Study it as though, but you'll know you have a cheat sheet backing you up. But if it's not important to you, that's okay too. So there's a quiz Tuesday on chromosomes, alleles, chromatids, homologous, homo and heterozygous. Hi. Yes. And I'll give you back your quizzes as well, so you can do corrections or, you know, like make additions to your cheat sheet if you need them. Oh, that's a good point. I will find it for you. Yeah, I'm just gonna look that up and put it on the board. Yeah. Thank you for asking. I've had a couple requests now. Yes. Yeah.
A lot of people left already. They're
And that's going to be on pages. Thank you. Oh, thank you so much. Yes. Honestly, that one looks like an animal cell, so I'd say that's what's on there. Have the 2 lines. It should all set on line. I'll finish this in a moment, so just be patient for that. Yeah. Okay.
It's Ben.
Hi.
Why is mine out of 22 and hers out of 20?
Because I'm a moron. It should be out of 20. So this would be 19 out of 20. There you go.
Thank you.
So sorry.
That's lower than 21.
But I wrote it down in here as 19 anyway, so it doesn't matter. I don't know why I didn't change it there. Okay, pages are up. Pages are up for what the quiz on Tuesday is on. Okay? I'll also post it up on Teams.
Progress report before the end of the year?
Yes.
Okay.
How about I try and get one to you, like, I can work on putting that together for you by the end of today.
That'd be great.
And maybe it'll be ready by the end of today.
Okay.
And maybe it'll be ready by Monday, but it'll definitely be ready by, yeah? Yeah.